Hello, my name is Madison Hawk. I will be talking about the cellular level of organization today from Module 3. So first we're going to start off of the structure of cell membrane. The cell membrane acts as a selective barrier separating the cells inside from its surroundings. It's made of phospholipids, cholesterol, carbohydrates, and proteins. We describe this as a fluid mosaic model because it's flexible and it's dynamic and it can move parts around. Next is the passive versus active transport. So the passive goes with the flow and active will go against the flow. So for passive transport, this goes from high concentration to having a low concentration without using or needing any energy for this. And then for the active transport, they need the ATP or an electrochemical gradient. And then a good example for this would be the sodium potassium pump. Next is osmosis. Osmosis is a water movement based on high solute concentration. Low solute means water flows in, and then this makes the cell swell. And then high solute makes water leave, and this shrinks the cell. And then obviously you have the equal solute, and it keeps the same. So you can see down there, that's the real names for it, but I like to just remember it by this. Next is bulk transport. This is cells can move in large materials in bulk. Endocytosis brings things in like nutrients and particles, and then you have the exocytosis, and this sends things out like waste or cell products. So exo out, endo in. Great way to remember that one. Next is cytoplasm and organelles. The cytoplasm contains organelles with specialized jobs, like you have rough and smooth ER. And this just makes proteins or lipids. And then you have the Golgi for packaging, lysosomes for digestive, and the mitochondria for energy. And then you also have the cytoskeleton for structure and movement. Next, we'll be talking about the nucleus and DNA. The nucleus holds DNA and controls the cell. DNA has base pairs, rules, and the nucleus inside makes ribosomes. Next, protein synthesis. Protein synthesis starts with transcription in the nucleus, then translation in cytoplasm. This is where tRNA brings amino acids to build proteins. Next is the cell cycle. The cell cycle includes the interphase for growth and DNA replication, and then mitosis or meiosis for division. So you can see there I put this chart or this picture, and it goes into a lot more detail and helps you understand about the G1, the S phase, and the G2, and then all of the prophase, metaphase, anaphase, and telophase from that. So then now we have the DNA replication. DNA replications happens in the S phase. And this creates an identical sister chromatids for cell division. Next, we'll talk about the mitosis. This is what I said earlier about the 5 phases. And this ends into identical daughter cells. So you have prophase for chromosomes forms, metaphase, line up in the middle, anaphase, they're pulled apart. Then you have the telophase, this is where the new nuclei forms, and then cytokinesis, this is where a cell splits. So you can see there I put that great demographic there. So you can see how it starts just the 2, and then you can see them forming, and then by the end, cytokinesis, they actually split apart. So it goes into a lot more detail understanding and explaining that. Next, cell differentiation. This is how cells take on specific roles with stem cells, and they're able to become any type. Lastly is, this is what I found from the book that I found interesting. This is about the lysosomes and cellular self-destruction. So lysosomes can do more than just recycle parts or cell parts. In damaged cells or in healthy cells, they can release their enzymes into cytoplasm, and this can destroy the cell. This process is called Autolysis, it's a type of apoptosis. It's a natural and sometimes necessary process in development for removing any faulty cells. So you can see there, I put a little graphic also on this one, so it goes into more detail and explains this. That's all. Thank you.